All right, welcome back. I am Jesse. I am a tutor in Melbourne, and this channel is mostly focused on doing GAMSAT videos. Today's video will be kind of like another sample questions walkthrough. So I'll upload the questions. They should actually already be uploaded to the resources page, which I'll link below as well. This will be a bit of like a worked solutions video, but it's more about going through the strategies that I might use to break down the questions. Hopefully they come in handy. Today, what we're going to be doing is going through more biology related questions. For the most part, people find these a fair bit easier than the chemistry and physics questions, which we're probably focusing on a little bit more here, but the bio ones do have the potential to be tricky. They're very qualitative a lot of the times. There can be a lot of just really tricky, wordy questions, and in the past week or so, we've been looking a lot at kind of reading strategies, ways to break down questions, and I think biology questions really lend themselves to that kind of practice as well. So also as well, if you haven't already, do subscribe to the channel. It really does help. It helps the videos get seen more and more. And from the feedback I've seen so far, people say that these are pretty helpful. So I'd like to have them reach more and more people. So spread it around if you can. Okay, so the first two questions are actually the same as the ones that I went through in I think it was my last video, or the one just before this maybe, on reading strategies and things, where I looked at note taking. So what I'll do is I'll just now put up the explanations from that one. All right, so here we'll go through it. So hormone sensitive lipase is an enzyme that in humans is encoded by the LIPE gene. And so in terms of notes and things like that, right? Obviously I would have the scratch paper. What I might do is just create a little segment down here to pretend like this is the scratch paper. And from here I would just go, okay, HCl, that's all I might do. Or I might even do like LIPE gene to HCl, understanding that, all right, maybe the gene to the protein is important. I'm just going to write that down. HCl is an intracellular neutral lipase that is capable of hydrolyzing a variety of esters. So there we go. So it's breaking ester bonds. The enzyme has a long and a short form. So I'm going to put L and S like that. And you might have different notation styles. And then it says the long form is steroidogenic in steroidogenic tissue such as the testis, where it converts cholesterol esters to free cholesterol. So here I'm just going to put long form, and I'm just going to put testis here, and then into, and then cholesterol, CHOL. And then it says the short form is expressed in adipose tissue. So I'm just going to put the word fat instead, where it hydrolyzes triglycerides. So and that works fat tissue and triglycerides fat into fatty acids. I'm just going to put FA or FFA for free fatty acids. And then HCl HSL functions to hydrolyze either a fatty acid from a triglycerol molecule. So I'm just going to put like a little thing like this, like an E, so a glycerol with 3 fatty acids coming off of it, freeing a fatty acid and a diglyceride. Perfect. So that means it's cutting it like this into a diglyceride and a free fatty acid, and that is where HSL is coming in. So I'm mapping out the pathway because it hasn't been given in the question. Then it says this is in mammals, another enzyme found in adipose tissue, ADGL. I often don't read the actual name, I just go straight to the acronym because they're probably just going to use that. Has a higher affinity for triglycerides than HSL and predominantly acts as the enzyme for triglyceride hydrolysis in the adipocyte. So I'm going to put this, it does the exact same thing, ATGL. I'm going to circle it, that one's better. Out of the two, it has a greater affinity. But then it says after that, HSL is also known as a triglyceride lipase, while the enzyme that cleaves the fatty acid in the triglyceride is known as diglyceride lipase, and the third enzyme that cleaves the final fatty acid is called a mono. So it basically keeps going, and I'm just going to put the word di into a single and another free fatty acid, and then mono, like that, and then should just have 3 fatty acids at that point and a free glycerol. And then now I've got the full pathway mapped out. Only the initial enzyme is affected by hormones. So I'm going to put H for hormone, and therefore it is the rate limiting step. So I'm going to put rate limiting step. The triglyceride and monoglyceride enzymes are 10s to 100 times faster. I'm just going to put times 10 times 100 like that. I mean, obviously either of them could be times 100 and so on. but it's just to put everything down. And I would do this often, like just kind of scribbling, not really looking at the notes a whole lot. They'd be quite messy, but it just helped me engage with it as well so that I didn't have to go back and reread. This is probably overkill for the notes, but it's more so for the emphasis or the kind of explanation or demonstration of how it works. And then the HSL is activated when the body needs to mobilize energy stores and so responds positively to catecholamines or ACTH. So now we're going to put in, I might do it like this because I need more space. I'm going to put a little positive with ACTH. It's inhibited by insulin, so I'm going to put a little negative insulin. And then previously glucagon was thought to activate it. However, it's the removal of inhibitory insulin that actually activates it. So I'm just going to put here remove equals plus like that because it's too awkward to kind of get it onto that. I've gone outside my bounds there. And then the lipolytic effects of glucagon in adipose tissue is minimal in humans. So basically glucagon is not relevant to humans in that case. Then another important role is the release of cholesterol from cholesterol esters for the use in cholesterol esters for the use in production of steroids and cholesterol efflux. Activation of the HSL is important in ameliorating the generation of foam cells in atherosclerosis. So when I read that, I kind of go, that's a lot of jargony stuff. It's relatively short. I've got enough notes. If I need to, I'm just going to rely on that. That prevents me making too many notes and killing too much space. So question 1, I've written here, so a mutation in the gene responsible for the production of which molecule is least likely to cause significant impact in the triglyceride metabolism in adipose tissue. So what I'm doing here is looking for word association. And I'm kind of going, all right, where did I see adipose tissue? So first of all, I looked to my notes and that was fat, and I could see fat was here. So that is short form. And then I go through the scenarios and I'm just going to find where each of these are and then consider their impacts individually. So HSL, that was a rate limiting step. So a mutation there actually would have an impact because that's determining the rate at which metabolism occurs, whether it's increase or decrease. So immediately that one's out. ACTH was a promoter or an excitatory relationship between ACTH and HSL. So it upregulated the HSL. So therefore, if we had an impact there, that would then mean that you wouldn't have HSL activation. And so since HSL is impacting it, anything that impacts HSL is also directly impacting it. And so that's our ACTH. Then ATGL, very, very similar, was down here. This was an alternative enzyme that could also help in the hydrolyzing of triglycerides in adipose tissue. And so this said which has the least likely. So this is a direct contributor to it. So that one's out. So by elimination, I've got glucagon, but I would still look for supporting evidence. Glucagon was minimal in humans, but this didn't say in humans, so it's still open to all mammals. But it did also say that glucagon was thought to be activating. However, it's actually the removal of insulin that activates it. So therefore, this was thought to be the problem or thought to be the contributor, but it's actually not. So the answer then becomes D. And you can see that I didn't have to go through all of this, right? I just look to my notes each time. And so as long as they're barely, like if they're kind of being done as I read, then it's really not taking up any more time. And then question 2, so consider the following statements. And then what I would do is skip them, go straight to the prompt, see what I'm looking for instead of just reading them. Which of the statements are correct? And I'm just going to circle the ones that are correct and then work out my answer. So HSL would be inhibited immediately following digestion. So I didn't see anything about digestion in the reading, right? And I've not mentioned anything with digestion. So I go, all right, well, where does that fit in? And I look through and I go, well, insulin would be released immediately after digestion in order to control glucose regulation. And so then what was insulin doing? That was inhibiting it. So it said HSL would be inhibited immediately following digestion. That basically means insulin release, and there it is there. And that's that kind of translation concept. So 1 is definitely right. So it means D is out. Then 2, the long and the short form of HSL catalyze the production of equivalent products. So we had long and short, and I look to see what they do. This one makes cholesterol, and this one here makes free fatty acids. Technically not equivalent, so that one is out. Then from there, so that means 2 is out, so A is out. And then 3, the removal of insulin has an excitatory effect on HSL. So we look to where that is, and it says removal. It does say plus. This is where my notes may have actually led me astray a little bit here. The keyword is excitatory. So excitatory and inhibitory are completely opposite terms. So it's really just removing the inhibition and allowing a reaction to take place. It's removing the blockade, so to speak. What it's not doing is promoting it in any particular way. So it's not actively promoting it or exciting the process. So it's actually not true, and so therefore C only. So you do have to always reason them out, and if you're ever unsure, always check back on the actual information if you're questioning it, and this is how they can throw tricks into the questions as well. All right, and then I'm going to try and keep the questions on screen, but you could also, if you wanted to, jump to the resources page and open that up if you want to split screen it or something, or if you've got this running on your phone and have this up on the computer or vice versa. But I'll try to keep it all on screen as much as possible. Cool, so Unit 2, so we've got research into the neural pathways of skeletal muscle contraction has led to the understanding, and I noticed that my typing is, I have so many typos, understanding of its origins as being traced to the motor cortex in the brain, initiating the acetylcholinergic or intrinsic pathway. So let's look at a bit more note taking. So here I'd be looking for any definitions like I talked about in the previous video. Is here it's talking about acetylcholinergic, also connecting the word intrinsic pathway, and this uses the depolarization of acetylcholinergic motor neurons. That's all I really care about, because it's already said acetylcholinergic before, secreting acetylcholine, that makes sense, into the neuromuscular junction of skeletal muscle. There's our connection, causing muscular contraction, done. Then we've got muscle stretch receptors, so MSRs. You can see whenever they use acronyms, they're often going to rely on using them, and so when I write these questions, you'll notice a bit of a pattern in the way that I write them. I often define acronyms and then use the acronyms or switch back and forth between the full word and the acronym itself in the questions. And this just puts a little bit of pressure on your reading comprehension, because people who have to bounce back and forth are generally going to run out of time. So you want to be looking for any defined variables, even if you're not familiar with them. In the tendons of the muscle measure, the relative, muscle stretch receptors, MSRs, in the tendons of the muscle measure the relative force of contraction and can have impacts on the motor cortex. Right, so we've got, I guess we'll put in force of contraction as well, impacts on the motor cortex, and they're calling this one, or I am calling in this one, the efferent skeletal muscle regulatory neurons is what it's using, and that was SMRN. And the enzyme acetylcholinesterase, we've got a new thing coming in, which is responsible for the degradation of, typo, that should say acetylcholine. Let's just block that out. So it should just be the degradation of acetylcholine in neural synapses. Right, so it's summarizing so far, right? I would try to avoid writing everything, but in my head, what I'd be doing is going, okay, so we've got an intrinsic pathway, which was acetylcholine onto the muscle from the motor cortex. Then we've got muscle stretch receptors, which are having impacts on that motor cortex. And then they're using SMRNs is all we're going to use for now. And then we've also got an enzyme that's breaking down the acetylcholine at the same time. So then we have new studies into the alternative excitatory pathway initiated by dopamine, or DA. So there's another one. Dopamine produced in the substantia nigra in the brain via electrical stimulation, which is now going to be the extrinsic. So that's really important, because now I can expect that there should be a comparison between the intrinsic and extrinsic pathways, have also been conducted, although acetylcholinergic stimulation of muscular contraction is separate from the dopaminergically, tongue-twisted, regulated initiation of muscular contraction that is impacted in Parkinson's disease. So we've got dopamine in Parkinson's. Even though this is separate, the experimenters have excluded participants with Parkinson's disease. Then it says figure 1 below is a visual schematic, and we've got little plus sign signifies an excitatory impact, minus signifies an inhibitory impact. And now here I've done my best to do a rendition of what I think might be the neural and hormonal map of it. So again, with this, I wouldn't really, as I've said in other videos as well, I wouldn't be analyzing this too closely. All I'd be looking for here is some of the key words, and I'd start to go, do I already have this information in my head? Is it summarizing something I already know, or is it providing something new? And if I kind of look around, I can see all the key parts and I go, oh yeah, there's motor cortex, motor neurons, there's the acetylcholine, there's a muscular contraction. So I go, oh, there's the, like, that there is the intrinsic pathway that was described, but I'm not going to actually do any more note taking or pay this any more attention, because I can know, okay, that's just summarizing all of the excitatory and inhibitory information. And so I'm probably going to go to that because I prefer to read visuals rather than words. And I could do the same thing even in reading the question. I might have even glanced and seen an image and maybe read a little bit and checked, and as I was reading, dropped down to the image and checked to see if it's summarizing, and then I could start to skip read and just look for the stuff that I don't see mentioned in the diagram. So for example, the stuff about dopamine, it's partly here, but then when it starts talking about Parkinson's disease, that is not represented. So that I would maybe read a little bit more closely. All right, so then we've got nerve blocks can be applied to a specific neuron bundle, and a summary of the available nerve blocks relevant to this neural network is shown in table 1 below. We've got that here, and we've got 4 different nerve blocks, what their targets are, and then what their minimum concentrations are as well. And again, I wouldn't be reading that too closely. I'd just be looking at the column headers to try and understand what information it's providing me. And I go, okay, anything to do with nerve blocks, I'll come back to that table then, because it's completely separate information from the diagram. So the first question is, which of the following does not have excitatory effects? So excitatory, inhibitory, I'm going to go to the map in that case. And I'm literally just going to test each one and just track it back through. I might have some memory of exactly what I was reading, but it's probably better to just check it all through anyway. So motor neurons, you can see there, that has an excitatory, it's got a positive, so that one's out. Muscular contraction, which I can find down here, has a positive effect on the MSR, so that one's out. And then muscle stretch receptor, which remember was MSR, so we can immediately find that here, and we can see that has an excitatory effect and it has an inhibitory effect. This question says does not have excitatory effects. Even though it's got both, it has excitatory, so that rules that one out. And I'm still going to check if acetylcholinesterase is right. So I find that here, and I can see it only has one connection, and it is inhibitory, so there's no excitatory. So then the answer is D. Relatively straightforward question to start it off. Going through question 4, if a study is testing the hypothesis, right? So we're looking for where we can measure the difference between having the substantia nigra working and having the motor cortex working. So there's 2 different ways. We could either play around with switching on and off the substantia nigra, but we may not have a nerve block available for that. But the other one that we could do as well is we could also be looking at switching off the motor cortex and allowing the substantia nigra to work. And if it does work, then we should get muscular contraction. So if we run through each one, nerve block A here, I already kind of know, basically if I block this, switching it on and off, and I can get a measurable difference, that's a possibility. And I could also maybe block this as well. And so if I look for which ones block that, nerve block A blocks the motor cortex, so that's a possibility. And then as well as that, we've got dopaminergic neurons, which is B, so that could work. And then D could also work because that's blocking the substantia nigra. Now you can't have 2 correct answers, so I kind of already just from that logically would go, it's probably going to be A. But to be exact, we can actually explain it. Nerve block C is not going to have any impact because that goes for the efferent SMRN, which goes this way. So that has nothing to do with this here. I mean, it is blocking the inhibitory path, but it's not deciphering between these 2 sources. So C is immediately out. Now with B and D, they're both, one of them is B is blocking the dopaminergic pathway, whereas D is blocking the original source of that. And in both of those cases, they should have equivalent effects on whether or not there is muscular contraction. So already they can't both be right. And the reason why they actually won't work is more so from testing your hypothesis between your experimental group and your control group. If we were to switch off either of these with this pathway and allow the motor cortex to run, then we get muscular contraction. If we then remove that nerve block and allow the substantia nigra or the dopaminergic pathway to join, it's joining in with the motor cortex, which means that even if it is having an impact, we wouldn't know because we're still going to get muscular contraction. If it's not having an impact, we also wouldn't know, because we'd still be getting muscular contraction through the intrinsic pathway overall. And so it doesn't actually help test the hypothesis, even though it is related to the dopaminergic path. And so therefore the answer will be A. So then number 5, which of the following would result in a positive feedback loop in the intrinsic pathway? So first of all, keywords, right? They often limit the question to a specific part, and then they put answers in that are correct but for other elements of the question. So sometimes the trick is actually in the prompting in the question itself. So we're looking for a positive feedback loop where we have effectively no inhibition, right? Negative feedback is where you've got inhibitory processes responding to basically slow down a cycle or to prevent it from continuing, whereas positive feedback is where you've got only excitatory loops and the pathway keeps getting stimulated successively. So which of these would result in that? So electrical stimulation of the substantia nigra, even though that says the word stimulation, if you stimulate this here with an electrode, then you do stimulate this pathway, but it hasn't stopped the inhibitory pathways here and here, right? So you're not actually creating a positive feedback loop. I'll get rid of all of that. The negative feedback loop is still present. You're just increasing the source of stimulus. So that one's going to be out. So with B, a mutation in the gene for acetylcholine transferase, a transporter protein required for the membrane transport of acetylcholine. So where did we see acetylcholine? We see it here, and it says a mutation in the gene that is allowing it to actually transport across membranes. So anything to do with its communication here, particularly probably its release from the motor neurons, isn't going to be able to happen anymore. So what you'd be doing there is removing this pathway and then blocking everything else downstream. And that includes everything connected to it. So what you got to be careful of is here is you're not even getting muscular contraction. So again, that's kind of messy. So if I kind of put a block here, it means all of this is cut off, which includes MSR, which actually also directly affects the acetylcholinesterase, which has the inhibitory loop. And you're also blocking this as well. So really you've only got this bit here that's outside of what I've circled. And so even though it's blocked all of the inhibitory pathways, it hasn't actually created a positive feedback loop because there's no positive feedback onto the stimulus in any way. And so B is going to be out. So then we have a mutation in the gene for muscular stretch receptors. So again, we can assume that that may have a negative impact. That's the more likely of the two, but it could be positive. A mutation in MSR, if we have a block here, then it means that we lose the inhibition. And so therefore, there is nothing then to tell the motor cortex to stop. So what will happen here is we've blocked MSR, so it means that we block the inhibition of the motor cortex. More importantly though, we block the acetylcholinesterase. So what I like to do in excitatory inhibitory pathways is just think of them as on-off switches. So if I switch off this switch, then it means that I effectively switch off the off switch, which is kind of like an on then. It's a double negative. So now if I've blocked this, then I don't have anything to break down the acetylcholine, which means that then I have a positive, well, not really a loop, but we'll say a continuation of the reception of acetylcholine because it's not being broken down anymore because the acetylcholinesterase that's responsible for that is now gone. So that's the closest we've got to a positive feedback loop. So that's looking most likely. And then D, administering of nerve block C of concentration 4 milliequivalents per liter. So we'll double check again. Nerve block C was affecting the efferent pathway. So again, that would be blocking the MSR pathway, which is good, but the concentration is too low. So the minimum concentration is 6, and this says it's only 4. So it won't actually have enough impact, which means you'll still have the MSR pathway working, and therefore you'll have some inhibition and a negative feedback loop. So by process of elimination then, answer is C. And so then in question 6, we have Parkinson's disease is a neurodegenerative disease that includes a fall in dopamine. So immediately what I'm thinking is, all right, that was mentioned at the end of the paragraph. So I'll probably go there and not bother with anything else. Include a fall in the dopamine in the brain and loss of dopaminergic neurons in the brain. So that's important because I saw dopamine. There we go. And obviously some of this you might already be familiar with, and this is what they do. They often choose things that would have come up in a science course. And so people with a science background may already be somewhat familiar, but always check that they're not providing anything new that you haven't actually learnt before. And also never rely on outside knowledge when it's super specific. Always go with the information presented to you. So which is the best explanation for why participants with Parkinson's disease have been excluded from the study? So we go through, this is again a very tricky question. It's somewhat using the bio, but again, it's about testing your understanding of experimental research methods and understanding what leads to the most valuable data, right? So A, they would be at greater risk of muscular spasm or seizure. So this one sounds like the kind of ethical answer, because it's like, well, if someone's got a disease, probably best not to experiment on their brain or anything when that's the part of the brain that is impacted by their very disease. But there's actually no mention of risks to them or anything. So we don't have any evidence. You might want to think, well, is there a possible risk? But we can reason that out as well. So Parkinson's is actually low levels of dopamine and low levels of dopaminergic neurons. So if we were to go back up and look at that study, what were we doing? We were stimulating electrically the substantia nigra. Now, the substantia nigra was actually promoting motor neurons here via dopamine. But if they don't have very much dopamine, then it means that you're not going to get much of an effect out of that at all. So if that's then leading to muscular contraction, if they don't have a very strong dopaminergic pathway, you can't actually stimulate muscular contraction very well. The other thing is that technically, and it's mentioned in the STEM, dopamine loss in Parkinson's is more to do with muscular contraction initiation and like fine motor movement and stuff. That's beyond first year biology though, so they wouldn't necessarily expect that, which is why I've written it into the question. So there's a few things that explain why there is some distance between Parkinson's disease and muscular contraction itself. But even if you went right into what the question says, you'll find that it actually will have a depressed impact, and so therefore it's not going to heighten the effect of muscular contraction. So seizure or muscular spasm is actually not the risk. Their results could undermine the extrinsic pathway hypothesis. So the hypothesis of the extrinsic pathway was, if we can electrically externally stimulate the substantia nigra, we may be able to get muscular contraction to occur in the absence of any effect of the motor cortex. So undermining it would be to suggest that it does not work, right? Or to suggest that it's not correct or that it's not supported. And this will actually be the most correct. Reason being is if I work through this, actually, I won't just give the answer. If I'm to work through this, I'd go, okay, well, they have less of this, I might use arrows, less of this, which means when you try to stimulate it, you still don't get much out of it. It increases, but you've got less than the average person, which means you've got less of this than normal, which means you've got less of this. Whenever you've got excitatory, it's just the same effect, which means you've got less of this, which means you've got less of this. And so therefore, if we were to use a Parkinson's patient, we would get less muscular contraction out of stimulation. Whereas if we used a healthy patient, then we would get much more. So in using the Parkinson's patient, that would actually be lowering or underestimating the effects of the substantia nigra potentially because of that limitation, or because of the lack of dopaminergic neurons in their brain. So that would actually be looking pretty good. Let's rule out the other two though. So they may be aware of the expected outcomes of the study. That one's just to check if people are guessing. There's no mention of like blind, double-blind studies or anything like that. And it also can't change anything because this is biological. So it's not a psychological study. So even if you're aware of what the outcome should be, you cannot change the way your brain works if it's being electrically stimulated in any way. And then D, they would not be able to produce measurable muscular contraction. So that would be assuming that they have zero opportunity for it. So one, they can produce muscular contraction through the intrinsic pathway anyway, but more importantly, through stimulation, it's just a loss of dopaminergic neurons. It doesn't mean no dopaminergic neurons. They would just have a depressed impact. So that one says not be able to, that's out. Often a good way to go through multi-choice questions is avoid any kind of absolute or extreme statements. They're usually too simplistic. The ones that are a little bit more complex or have contingencies on them are usually the better ones. And then heading into the last one. This one's a little bit shorter and a bit more of like a problem-solving type question in biology. So restriction enzymes, sometimes referred to as endonucleases, are a protein responsible for the cleaving of DNA molecules at specific base sequences or recognition sites. So they're like, technically this is from first year bio, but maybe if you're a bit rusty on it or if you're new to some of this stuff, then I'd be highlighting. So we've got restriction enzymes, also called endonucleases. They're cutting DNA at recognition sites. Then we've got HIND3, uses this sequence. However, it can only cut through the phosphodiester bonds of 2 successive adenine nucleotides and the hydrogen bonds along this recognition site. So looking at that, I'd be going, okay, well, it can only cut between 2 A's there, right? The other thing I'd be noting is that's only single-stranded DNA, and DNA is obviously double-stranded. So I may want to write down the complementary side as well. For now, I'm going to leave that off. Then we've got another restriction enzyme, AFE1, can only cut the phosphodiester bonds between cytosine and guanine when arranged 5 through to 3 in that specific order. So here we go, I might even just do that. That's the more important bit. And its recognition site is this here. So in terms of note taking, right, because you're going to be doing it on a screen, what I'd be doing is just writing down on the paper, HIND3, I'd copy this down because it's relatively quick, AAGCTT3, and I'd be doing this as I read, and I go, okay, cut there, right? And then the other one was AFE, 5' to 3', and a same thing, write down its sequence, AGCGCT, and it says that it cuts only through CG. So I look for where I see CG, there it is, and there's nowhere else it cuts there. That's going to summarize everything from here to here now, really, really quickly. So then restriction enzymes are classed as either A or B, depending on whether they cut directly through the DNA and leave flush or blunt ends, so that's class A, or whether they leave overhanging strands on one side of the cleaved end called sticky ends, class B. So we've got a couple of definitions. Again, you might already be familiar with this, but we'll note those down. And then it says which of the restriction enzymes is considered to be class A. So there we go, class A leaves flush or blunt ends. So I basically have to test each of them. So this is where now I would actually write out the sequence. So A, G, C, T, T, and we can use complementary base pairing, 5 to 3. This will run 3 to 5 on the other side, and we can just do opposites. So A runs to T, T, C, G, A. And we found that it cuts through A and through A, there and there. And then it can cut all the hydrogen bonds between them. So we can just run like that. And you can see it's making that Z shape or that zigzag. That would create sticky ends. So that is a class B. I'm not going to bother checking the answers. I'm just going to literally write them both out because the information that I get from this is probably going to be helpful in another question anyway. So then the other one is 5-3. Excuse the handwriting, it's pretty shocking. A, G, C, G, C, T. And 3' to 5', that would be T, C, G, C, G, A, like that. And it runs only through CG in the 5 to 3'. So we found that already here. And if we run backwards, we look for left to right, right to left. We look for where we can see C to G. We can see it just here, C and then G. So it cuts there as well, which is, get rid of some of that, directly underneath, like that. I've done it a little bit wonky, but it's cutting flush, right? So in a straight line. So this would be a class A, like that. And so which one is class A? That was AFE, forgot to do the labels. And this one was HIND. You can see I don't even really need to do the Roman numerals. I could call them H and A if I wanted to. HIND and ape comes to mind, like anything that just makes it easier to process. So this one was AFE only. And then we have half of a sequence of DNA is shown below. The double-stranded DNA molecule when treated with HIND3 and AFE1 would have what. So this one's a real pain, right? Basically, we would have to actually write out the entire sequence because you've got to do the 5 to 3. You could potentially, as a shortcut, you could kind of go, okay, well, if it's AA for hin 3, and then I would go, well, what's the complement to that? That would be TT. So anywhere I see AA, it's cutting on this side. Anywhere I see TT, it's just cutting on the other side in that case. And then for AFE 1, that one cuts CG. So therefore, on the complement, GC, right? So I can just look at the 5 to 3 on this one here and just see where I can find that, basically, going left to right. So I can see AA here, and that's all I can find. And then I can also see, well, I don't even want to do that actually. I want to look for the recognition sites. So if we do that first, a bit tedious, you can use your finger or something on the screen to kind of like hold it there, the edge of your scrap paper or something. And we want to look for AAGCTT. So we're looking for it, AAGCTT. So here it is here. We'll just do that. Look for it again, AAGCTT. And I can't find it anywhere else. So that's one site there. So I've got HIND, I'm just going to put H. And then I'm also looking for the recognition site of AFE. So again, 5 to 3, AGCGCT. I like to do it in trios. It's a little bit easier to remember. So I look for AGCGCT. Almost there, but that was already the HIND one. And here it is here. Nope, that's not it. Here it is, AGCGCT, like that. So that one's the AFE. So now if we just kind of zoom back into where we were, we could write out the complementary strand and find all the parts. Or really what I could do is just go, well, this one was a sticky end, so it cuts here, it'll run across and it'll cut out on the other side of TT because that'll have AA as well. And then for this one here, we knew this was a blunt end, so we just look for CG and we know that it cuts right across on the other side. And so from that, I can very, very quickly see all of my pieces. And so really it's just like blocks, like this. And there we go, right? And so I can see a couple of sticky ends here. Remember, they would all then separate because they're being cut apart. So I can see, first of all, there are 3 fragments. So C and D are out. It's down to how many blunt ends there are. I don't have to worry about the sticky ends. So you can see that the ends here they look blunt, but we've only got single-stranded DNA. And so we can't assume that they are blunt ends. They could have been cut from sticky ends as well, or from class B restriction enzymes. So we can only work with the 2 ends that are formed here. The other thing is technically ends are formed by a restriction enzyme. So we don't know the origins of this fragment, where it's come from, whether it's the full length, if it's from some particular organism. That would be overthinking the question. So we're just going to go with what cuts we get. We can see we get 2 sticky ends, and here we get 2 blunt ends. And so then that will be answer A, like that. You can see I made a few little revisions to the question, because as always, I write these things and I should probably draw them out and actually go through them. I do them in my head. And so I should probably check them a little bit more, because a few people have called out some errors on them, which is good. Keep doing that so that we can improve them. But I'll have those changed in the actual document as well. Cool, and there we go. So you can see that a lot of it actually just became problem solving in that last one, and it wasn't really relying too much on the reading. Obviously, if you're not from a science background, that may be a little bit heavier in terms of the reading. But also you can see there's very little maths involved in biology questions. They're mostly to do with qualitative analysis. They often give you scenarios that you're really not familiar with as well, outside of like genetics, but they often try to throw you off because the difficulty, I think, is generally a little bit lower than the chemistry and the physics for most. And they also usually use the biology questions as an opportunity to test knowledge of the empirical research process and that kind of thing. How do we create, valuable data and test hypotheses and that kind of thing, which they're just a pain sometimes of a question. You can work through them though eventually. Cool. All right. So yeah, that's another one there. Let me know your feedback on them. I'll keep trying to put more and more up. I'll have this added to the resources page. As I mentioned, it's starting to grow a little bit more, but if there's something else you'd like to see on there, you can let me know, because I'm pretty much kind of doing it to request. I'm slowly building up just a big bank of questions. All right, easy. Well, I'll leave that one there, and then I will see you in the next one.